science, academia, and healthcare. Please welcome me in welcoming our invited dignitaries, Professor L.S. Shashidhara, Director, National Center for Biological Sciences, Bengaluru. In his absence, Dr. Sharath Anantamurthy, Honorable VC, Kuvempu University. Professor Rakesh Mishra, Director, Tata Institute for Genetics and Society, Bengaluru. Professor Ullas Koltur Sitaram, Director, CDFD, Hyderabad. Professor Kumaraswamy Tangaraj, Senior Scientist, CCMB, Hyderabad. Join me in welcoming our special invitees, esteemed professors and guests from all across India's leading institutions, Dr. Pallavi Kshetrapal, Associate Professor, Delhi University, Dr. Kasturi Mitra, Associate Professor, Ashoka University, Delhi, Dr. Vijayshri Yellappa, Team Lead, Netherlands, Dr. Akanksha Chaturvedi, Scientist, BRIC NCCS, Pune, Dr. Shraddha Karve, Associate Professor, Ashoka University, Pune, Dr. Madhura Kulkarni, Senior Scientist, Pune, Dr. Radhika Nair, Faculty at CHD, Bengaluru, Dr. Mansi Rajendra Malik, Senior Scientist, TIGS, Bengaluru, Dr. Rajesh V. Iyer, Senior Scientist, TIGS, Bengaluru, Dr. Kumaraswamy HM, Professor, Department of Biotechnology, Kuvempu University, Shimoga, Dr. Nagaraja, Senior Professor, Kuvempu University, Shimoga, Dr. Tippeswamy NB, Professor, Department of PG Studies, Kuvempu University, Shimoga, Dr. Pandarish MD, Associate Professor, Adichunchandgiri School of Natural Sciences, Dr. Sham Sundar, Reader, Department of Oral and Maxillofacial Surgery, JSS Dental College and Hospital, Dr. Anand Kavde, Head of the Department, KEM Hospital Research Center, Pune. We also welcome the administrative team and management, dear faculty members of Subbaiah Medical College. As we commence this auspicious event, it is customary to invoke divine blessings by lighting the ceremonial lamp, a tradition that signifies the triumph of light over darkness. I request our esteemed dignitaries to kindly come forward to light the lamp and mark the beginning of today's program. I request the dignitaries to kindly stay back on the stage to inaugurate the Subbaiah Translational Research Initiative by cutting.
A very good morning to all. It gives me immense pleasure to have such research stalwarts under one roof to help us shape this translational research initiative. I would like to welcome Dr. Shashidhar sir, who has been instrumental and invaluable. His mentorship has been invaluable in today's gathering, to make this gathering possible. Dr. Rakesh Mishra, I met him yesterday and last evening we had a nice talk together, and I welcome you, sir, also. I welcome Dr. Vijayashri, a close friend of mine since a long time, and I also welcome all the delegates present here. There goes a saying, when you're passionate about something, the whole universe gets along to make things happen. And today's gathering is something like this. So I thank all of you all to come all the way from different parts of the country and from premium institution to be here. And without Dr. Shashidhar's mentorship, this wouldn't have been possible. Thank you, sir. I would like to just mention something about our institution, our journey so far. Our institution started in the year 2003 under the banner of Tadikila Subbaiah Trust to impart medical education and healthcare to all the areas which needed it the most. So we started with, actually the seeds for this institute started long before 2003, in 1995, when we started a small hospital, big then of course that time, Subbaiah Hospital in the city. Under the banner of Tadikila Subbaiah Trust, we started our medical college with 1,100 bed teaching hospital and around 200 PGs, UGs, and more than 100 postgraduates joining us every year. We also have a dental college with 100 students joining us. 350 students of the nursing are joining us in our nursing college, as well as allied health science and a physiotherapy coming soon. So these vast institutions give our UGs as well as a PG as well as our young doctors a good exposure to a lot of clinical patients, and they get an opportunity to treat a variety of patients. But it does not stop here. Like, only academics is not enough for any institution to grow. So we started our, you need to have something little different, like a lateral thinking. And with that idea and vision, we started a Subbaiah Research Institute, which is a strong vertical of the Subbaiah Trust, of the Tadikila Subbaiah Trust. Once we had our... So after that, we started with, the way forward, we went with our collaborations. And we are proud to be collaborated with premium institutions like the NCBS, the TICS, CCAMP, as well as PHFI, and a lot many institutions, and even international, with international universities. Recently with even the IIHMR as well as CCDC in Delhi. So we have these collaborations, and we have great outcomes also of this. I am proud to announce that we have received a grant from DRDO on KFD by Dr. Akanksha from NCCS Pune, who is with us, and I congratulate Madam, and Dr. Debashi from ICMR Bhopal, and our Dr. Balu from our institution. Also happy to share that Dr. Mansi Malik from TICS and Dr. Ramya have already started working on a project together. So these partnerships allows us to dream bigger and takes our institution to the next level. So today's meet is a wonderful example of such a collaboration. We recently, why this translational research initiative? Recently, we had a visit to NCBS as well as Coimbatore University. We were amazed to see cutting edge, actually groundbreaking research happening there. And that's when the idea about the translational research initiative came up. With a strong patient base and clinical expertise, and with a research inclination, we could help translate discoveries from from bench to bedside and again to the community. So hence, we have gathered here for this translational research initiative. I would like to just tell a little about the research we have been doing so far. we have started our research with few key verticals like the NCD, where diabetes and our clinicians are working constantly on doing research in NCDs like diabetes, cancer, and metabolic diseases, and to AMR as well as infectious diseases, nutrition and genetics, artificial intelligence in healthcare, biomedical and material sciences, clinical research and trials, and our faculties are using AI tools to screen for breast cancer, anemias, lung diseases, and to predict the outbreaks of dengue and complication of disease. And we are also in the process of developing our own Subbaiah cohort. So this would strengthen the community-based research and also add value to our institute. These are definitely early steps, but it reflects our determination to build a meaningful research culture, and this shows that we are headed towards a research that directly involves the community. I would also I feel also proud to see that a lot of students from our institute, the medical students as well as the postgraduates, showed interest in doing research. So last year, early this year, we started with a Subbaiah Student Research Council to provide undergraduate a platform to explore research and share their work. And we also had our very first UG conference, the named Cognitia, where we had more than 200 students participating all over the state. Our students also have been selected for an Indian Institute of Science IISc internship, where they got to interact with eminent scientists and see the lab work. And Dr. Shashidhar sir has agreed to allow our students for their elective postings in the third year as an internship in NCBS. Thank you, sir. We are also proud that year after year, more and more students succeed in securing ICMR projects. So our roadmap ahead, what we would think is this framework development meet, which we have inaugurated today, is a key step forward. It brings together clinicians, researchers, collaborators to design a structured roadmap for translational research. Moving forward, we want to identify or focus onto the key areas of research with strong clinical and societal impact, develop clear protocols and shared resources to support collaborative projects, expand training and capacity building to prepare the next generation of researchers, and strengthen partnerships between academia, industry, and the community to turn innovations into real-world solutions. With this, I would like to conclude and warmly thank all of the distinguished guests traveling all the way to help us shape this initiative. My deepest gratitude to all our faculties for their dedication, to our students for their enthusiasm, and also to the collaborators for their trust. Above all, I extend my heartfelt thanks to Dr. Shashidhar, not only for his mentorship, but also showing us what true scientific leadership means. And let me close with a thought. With your partnership, we build a legacy where science does not stop at publication, but reaches the bedside, the village, and the community that needs it the most. Thank you. Thank you, everybody.
Thank you, ma'am, for providing us an overview of Subbaiah Research Institute. Now I request Dr. Nagendra S., President, Subbaiah Research Institute and Medical Director of Subbaiah Medical College, to deliver the presidential address.
Good morning, everybody. By remembering the great visionary advisor who built this trust institution and made a great contribution to the people of Shumoga district. My father and the managing trustee, late T. Subbaiah, with his blessings, I will start few words for this inauguration. First of all, on behalf of our institution, faculty, trust and all the students, I welcome all the scientists, delegates and also our faculty members and all the people who are attending this research related discussion forum. As you all know, learning is a basic thing and taking that knowledge to higher level is by various means. Of late, after the last 3-4 decades, research has become the most important and fundamental things in all the fields. It could be in science, it could be literature, or it could be in the defense. Recently, you must have seen the performance of Indian Army in the latest war, where without involving any soldiers, by only using the latest technology, how India surprised the whole world. In the same way, in our medical field, we always look for research, learning. But of late, we realize that the clinical research, the expert scientists in the medical field need to be associated with various other scientists in other fields. It could be in physics, chemistry, various things, so that these type of things, with the interaction between we can come out with the best of the best. So our research institution, now we are trying to do a lot of good work with our expert team like Dr. Balu sir, Dr. Vikram, Dr. Ramya, Dr. Shubha and all other related things. And of course, I remember Dr. Kashinath, Professor of Biochemistry, in the early stages of last 10 years back only, he started small research activities and he was requesting me to publish papers and he was trying to do something, so that has taken us in a big way. I did my MBBS MD from Manipal, where I used to see everywhere, wherever I go to preclinical section, clinical section, hospital, everywhere in 1980s and 90s, I was sensing that importance what they gave to the research has taken them to very higher level actually. So we are proud from Subbaiah Institute of Medical, Dental, Nursing and Paramedical Sciences that our team is doing wonderful things. Definitely it requires the blessings of all of you, Dr. Shashidhar and all other, everyone has directly, indirectly contributed and helping us throughout Indian this thing. By seeing the delegates, maybe it is the first time or a first of its kind, these type of great people are coming under one roof and a institution like ours. So I am very thankful to you, proud of you, and I wish all the best for the discussions which you are going to do, interactions you are going to do, and the knowledge which you are going to give us. And also kindly consider our institution in any of your contribution, any of the association. We are ready, so you can please advise your friends, your well-wishers and all the peoples at different levels in the country that what initiation we are doing, like you guide us to go in the right direction. I wish all the best for this research initiative framework developmental meet to be held here actually. Thank you very much.
Firstly, I would like to thank our chairperson for making my job easy. She has mentioned all the verticals and most of the research we are doing. So our basic idea is to start a sustainable translational research with main focus on mother and child health. And we are also looking at point of care diagnostics, which are affordable and easy access. And currently, as ma'am already mentioned, our verticals are mainly on nutrition and genetics, infectious disease and antimicrobial resistance, artificial intelligence in primary healthcare, and One Health, and material sciences. So as ma'am already mentioned, we are already collaborating with NCBS and TICS, and we have started quite a few projects, of which few are on hepatitis B virus and multi-drug resistant Acinetobacter baumannii, and detection of Salmonella typhi from blood directly. So basically our strengths lies in our patient load and also our people, our ecosystem, which helps in developing the various research initiatives which are planning to take up. So the basic strength is a pediatric cohort. We have a large number of pediatric cohort and also adult cohorts with acute respiratory infections, neuroinfections, arboviral infections. And we have a MDR repository also, and critical care patients. And we are planning to do research mainly on infectious disease and nutritional problems. So we have one of the most important vertical, that is artificial intelligence in healthcare. We have a Center for AI Research and e-health. Under this program, we are running lot of disease screening in rural areas, mainly on diabetes, breast health, cardiac health, eye, lung health, and anemia. One more most important thing we are doing is KFD. As we all know, Shumaga is endemic for KFD, and Western Ghats, we have a lot of arboviral infections. So we have completed KFD serosurveillance along with ICMR. And as ma'am mentioned, we have a recent project on monoclonal antibodies development against KFD non-structural protein 1 along with DRDO, that is Dr. Akanksha. And we are also working on adolescent and women health initiatives, which mainly focuses on nutrition and menstrual health of the women. So the next vertical which we are mainly focusing is nutrition genetics. So under this we have adolescent health and vitamin D screening, lead level detection in maternal and cord blood, and we have vitamin D3 detection. Also we are collaborating with Kuempu University on genetic studies, that is study of ICAM mutations in neurodivergent kids. Infectious disease, mainly we are looking at group B streptococcus, occult hepatitis B, dengue virus, pathogen typing, and focus is on rapid diagnostic test, that is point of care test, which will be affordable, and newer techniques for drug resistance detection, and a few areas on infection control also. We are also working on advanced biomaterials and regenerative therapeutics. Under this, mainly we have phyto-assisted synthesis and advanced characterization of pure and doped metal oxide nanoparticles with focus on their biomedical applications. And we are also working on small animal model for toxicological evaluation of nanomaterials and herbal therapeutic agents, and integrating clinical surgical experience with laboratory-based research for developing tissue engineering materials for neoangiogenesis and osteogenesis. And these are the other research under this biomaterial vertical. We have a blood lead detection by newer methods, semiconductor nanotechnology for hyperbilirubinemia, nanotechnology which helps in wound healing, and biomedical waste valorization on gypsum management, and also nanoherbal formulations. The other area is non-communicable disease with main focus on diabetes with our endocrinologist Dr. Praveen sir, who is working on type 1 diabetes patients with a large number of database, and also type 2 diabetes, and we have a clinical trial running on the glycemic control of newer drug molecules. Also sir is working on disorders of sex development using genetic studies, thyroid malignancies also. And we have a endocrine surgeon who is not here today. He is working on parathyroid autotransplantation, and he is also working on inflammation score for thyroid nodules, oncoplastic techniques, and he is working on a genomic landscaping of endocrine and breast malignancies, planning to create a biobank for breast cancer tissues. And we are also exploring into newer molecules for elderly health, of which cartilage oligomeric matrix protein for knee osteoarthritis detection, and serum estradiol and testosterone levels which are associated with hip fractures in elderly men. Our anesthesiology department is also working on few of the models, that is the propofol models, nerve blocks, anesthesia cytokines, and post-operative nausea and vomiting treatment. Ophthalmology department is mainly focusing on cataract, pediatric ophthalmology, and squint. Quite a few studies have been completed and they are planning to do few more. Our dental college, they are working on oral malignancy, that is utilizing optical multispectral screening device for non-invasive detection of oral potentially malignant disorders and malignant lesions. So these are the key research areas which we are working on too, and these are our collaborations which we are having right now. With further support from all of the dignitaries who are present here, we would like to collaborate with the areas which we are interested in and extend our research focus. Thank you.
Nice week. You are okay? Okay, good. So I'll not get into the any formalities. Thanks for organizing in a very simple way without much garland and nothing of that kind. So there is actually a neuroscience meeting that NCBS, ISA Pune and others organize. It's itself called No Bouquet, No Garland meeting. It's named as No Bouquet, No Garland meeting. It's good to have such meetings. Okay, thank you very much to all my colleagues, friends, collaborators, both from Shimoga and from outside who have come here, and all students and faculty here. Just hearing the overview of the kind of work you're already doing, you're already on the right path related to research. So there is nothing new that we need to add in the context of why we need to do research. But I just, for the understanding of younger people over there, just mention when we talk about research, medical research, there are three components. We call it as health research rather than medical research. These earlier we used to call medical research more often, translational and medical research, but these days we would like to use the word health research, because health is not only related to what happens in the hospital, right? It's also what happens at home, what happens in the society at large, how we keep our water, how we keep our soil, how we keep our entire cycle of the food cycle and keep the environment, and then how that prevents and reduces the health burden in case, because of unfortunate reasons like genetic background or because of some other, like cancer, for example. It's simply an accidental mutation in somatic cells and it may lead to cancer. So there's nothing that, irrespective of how healthy one person is, anybody can develop cancer, right? Similarly, there can be accidental reasons of variety of other reasons, stochastic reasons. Someone may develop, diabetic. But country like ours, with so much of genetic diversity, geoclimatic diversity, and nutrition diversity, there are huge variety of different other kinds of diseases by which there is a big burden. India is one of the largest health burden countries, and we need to reduce that one. And to do that, of course, we need to do much more work in a hospital like this, attached with a medical college, because one is you have much more diverse specialities available, and students and postgraduate students who wanting to do research, and they can actually provide inputs, they can provide human resources that is required to do community-based research, cohort-based research, and so forth. That understanding, when it comes to lab, will help how to reduce, how to prevent, and for that you need to do diagnosis, you need to develop very different screening methods, for need right indicators. The right indicators will come when you actually screen large number of people. Whether it is the invasive indicator or non-invasive method of diagnosis, or using molecular markers, using some other method, we need good, low-cost, high-throughput screening methods first to understand the diseases, its prevalence. And for treatment, we need better diagnosis, more precise diagnosis. right? So for that, we need to do more customized kind of diagnosis, and that leads to more customized and personalized treatment for treating the disease. So when you look at understanding the health, understanding the health burden, understanding the very different factors that influence the health, and reasons for which where things can go wrong, and people may suffer with one or other diseases, and then how to diagnose. Diagnosis for the purpose of giving the right treatment, right? Diagnosis can also be part of. Screening is also nothing but a diagnosis, but screening is mostly to understand whether someone is positive, negative, someone is low, the high risk or low risk and kind of stuff. At some point, when they come to the hospital for treatment, with some symptom, clinical symptom, you need more precise diagnosis. So when you do research, you need to connect all these dots. And that's precisely a large research institution should be supposed to be doing, health research institution. And unfortunately, in this country, and with 1.5 billion people, with fourth or fifth the largest economy in the world now, we still do not have more than 10 good health research institution in a medical college hospital setup. We can actually, if you ask anybody who have traveled across, Dr. Balu or anybody who ask, what are the hospitals and the medical colleges which are also doing research? We can, in Bangalore, we can call St. John's Research Institute, in CMC Vellore could be another one, AIMS in Delhi maybe is another one, right? And KMC here, Manipal, But again, when you look at gradation, the top quality which actually can be compared to the world, right? Ulas is another expert who knows the kind of research that's happening in the health sector in the country and beyond. So we can it's very unfortunate that we have very few. There are a variety of reasons maybe. One is lack of resources, lack of expertise and so forth, right? Particularly if you're in a non-metro places, how to attract faculty, both teaching, clinical faculty as well as research faculty. How do you attract them to your place is always a question mark. But the modern methods of doing research and modern communication technology and our mobility, increased mobility and ease of travel, actually enables us to do good research. Good collaborative research. You don't need to have all the expertise in the same campus, right? You can actually collaborate. It could go to Bangalore, Pune, Delhi, Hyderabad, doesn't matter where the expertise is easy to collaborate as long as there is an sincere interest to do research in terms of collecting the data, analyzing the data, collecting the sample, analyzing the samples, and then sit together and interpret the results, and then see whether it leads to new diagnostic methods or new treatment methods, and how to do clinical trials. And all of these things can be done very well with the help of collaborators. So this initiative is to not necessarily, because there's nothing to inaugurate a research institution, you're already there. It is more about setting up a collaborative research initiative, you can call it as, because you can collaborate across the country with the help of experts. Some of them are here. There are many more in different parts of the country. Some of them are close, some of them may be far. But now with good mobility, it's okay. And only Tangaraj had to travel 7 hours for some reason. But Belgaum to Shimoga should have been 5 hours. I don't know why it took 7 hours. So I guess, that's one part, right? Why we are here, what we want to discuss. This is also to all my colleagues and friends who are here to sort of tell you that, please focus on the translational component and the collaborative component and what we can work together. And coming from the region where this kind of research has not happened, there is a particular health burden in this region too, which is similar to the kind of health burden which is there in the rest of the country, and some of them are unique to this place. Some kind of infectious disease like KFD is unique to this region. There are certain kinds of nutrition-related deficiencies. Malnutrition is also unique to this place, right? And there are certain kinds of cancer manifestation is also unique to this place. When it comes to dealing with AMR, the cause of AMR, if you want to prevent, right, is again, what is this antibiotic cycle that's going on in this part of the region, which is actually leading to increased incidence of AMR. One has to study before you actually look into what kind of intervention you want to bring in. So each region has to have its own innovation, its innovative solutions for which you need to do research, right? It's not like one solution fits for all. It's definitely not going to be effective both for agriculture and for health research, because we are dealing with living, organisms in agriculture with plants and in health with human, right? So obviously, there is every region, every medical hospital, every college had to have its own research. Without that, will not be very effective in giving the the support system for improving the quality of living, quality of health and healthy aging and healthy living, both in every part of the country. The three points I want to highlight, then I'll hand over to Rakesh. One is the data should be really precise. You don't have to worry about what is the quantity of data you're collecting, whether you're getting all the data points that you need to collect. But as much as possible, the people who collect data should comply with, whatever the protocol that is mentioned. And that data has to be digitized. Without digitization, these days we can't do multifactorial analysis, the kind of thing that you already have in artificial intelligence analysis sector, which is very important for this. Second is setting up a high quality and a large scale biorepository, which is most important. Madhura has set up one small one in Pune. She can help you, particularly for cancer. And Pallavi Kshetrapal is joining in half an hour or so. Have a discussion with her. She has set up one of the largest, and she's the one who is also involved in training other people to set up biorepositories and provides support through DBT and ICMR. And you should talk to her. Then these two are critical: data collection and process, and how you maintain the data in accessible and interoperable kind of databases, and also samples, right? And that those two are there. Then, of course, research projects, which will emerge, it will evolve, and then you will definitely find good solution with the help of experts. There are certain kinds of facility you need to have: clean room, clean air, clean power, and uninterrupted power. And the second is some basic preparative research lab required. For example, you have to isolate PBMCs and store. You have to have it to here. You cannot send the samples to Bangalore and say isolate PBMC. There won't be any PBMCs by the time the blood samples reach Bangalore. It has to be local. Then some basic genetic and genomic facilities like PCR and small NGS and so forth. And then some biochemical characterization labs, which most of them are already there for the diagnostic purposes in your pathology and your diagnostic labs. But you need to expand that kind of one for research purposes, slightly different grade. Very high-end, whole genome sequencing or NMR, or you have to do deep mass spec. Such kind of facilities can be with the help of collaborators. You don't need to set up all the facilities right here on campus. The second part, other than the infrastructure and the flow of collection of data and samples and safely keeping them with the ease of access, is ethics and policy and guidelines. Most important is to every individual who enters to a research initiative, a research center should have a thorough training in research ethics, research methodology and integrity, and also related to what are the rules and regulations of the country which they have to comply. There are certain kinds of consent they have to take. So ethics is the one which has enabled us to set up rules. Rules are not simply some bureaucratic set of rules. But rules, I'm sure many of you who are doing research already know. I'm actually addressing the students, not to Dr. Lata here. So, people say, there are certain, you have to take consent in a particular way, is a rule now. But why do you have to take consent is a part of the ethical question that you are addressing, right? So research ethics and certain kinds of rules and compliances are all sort of interdependent. And that's one thing we have to strictly follow. Without that, we will not be able to be truthful to the cause for which we are doing research. Research should not become an end of everything. Publication, not for the purpose of publication of a ranking, whether it's a NIF ranking or other things, it should be to solve problems. So to do that, all of these things are very important. Once it is done, automatically you will receive good support from everybody. The people would like to come and collaborate. People will bring their own funds, So funding should not be a problem if these things are followed strictly, because everywhere there is a demand for such kind of research initiative where the data is clean as much as possible. Data can be noisy because Indians generally don't report everything that they suffer. When you ask a patient very different questions related to the symptoms, very often they don't tell you everything, the background, what they're eating or what kind of lifestyle they have. So you'll always be noisy, right? But that's okay. They have to live with that. But once the data is collected and how you maintain, how you use it, that should be certain standard operating procedures, which will all attract more collaborators, right? And the last point is, of course, the translational. We should never stop at this point. Clinical trials, the variety of different types of clinical trials are possible, and that also is, there are set of norms and regulations. Funding is available. When from ICMR started giving funding for, the phase 1 clinical trials, right? ICMR gives for phase 1 clinical trials? Yeah. Yeah, you know that, yeah. I have to, yeah, yes. Yeah, that's true. So then the one last point, which is also translational in a way, is capacity building. All your medical college students, undergraduate, postgraduate, not only get trained, you also have to train if you're, supporting the healthcare service to this region, ASHA workers, and also the other hospital, microbiologists or pathologists or technicians working in the lab, and also government health officers. So we have a program, Rakesh and me are running in Bangalore, where we actually train, the pathologists to microbiologists to a very different technical, people from public health departments of Government of India, Karnataka, and very different districts. They come, and we train them in hands-on, molecular diagnostic, how to sort of detect, very different viruses or bacteria, how to detect AMR and so forth. We also train ASHA workers in local language, not only in Karnataka, and also in Andhra and in Telangana, where we also tell them, and also in Maharashtra. I don't know whether you attended one of those in Pune. PKC organized AMR workshop for ASHA workers. So to just tell them, they should be aware of what it means. When they go to door-to-door survey, they will also explain the importance of reducing the use of antibiotics. Because that has to come from them, because they are the ones who go to those houses and then see the people are suffering with one or other infectious diseases, continuously having infections like urinary tract infection, it never goes off. So these are the things where ASHA workers are the ones who will first find out. They also have to explain to the individuals in the house that what not to do, right? One of them is, of course, overuse of antibiotics. And they have to explain to them in a very simple way. So even ASHA workers also need to know, some basic concept. Obviously, they may not be able to understand all aspects of molecular biology leading to AMR. So I guess these are the things we can do together. We can organize series of training. I think we did one here, AMR awareness workshop, if I'm correct. Yeah. Okay, so thank you all. And one last point to my friends and colleagues here. Don't use the knife as sharp as possible to be clear that everything has to be done in a proper way when you want to collaborate. And don't look at this as a friendship event. This is not a friendship event. It is to solve a medical problem. Thank you very much.
Thank you, Subbaiah Research Institute team, Dr. Lata, and thanks to Shashi for coordinating this activity. And it's very exciting to see, and it also shows the intention of people that how we are all wanting to work together in a different kind of setting. So this is particularly important, and I'm very happy to be part of the history in which you are setting up a research institute of this nature in a smaller city with the ambition which involves institutions from many parts of Karnataka and other parts of the country. So Shashi has already explained and given really nice background of what this whole initiative is about. What I am going to do is to tell you how TIGS, Tata Institute for Genetics and Society, is relevant for this and very much willing to work with this kind of initiatives. So we are an institute with a vision to use cutting-edge science and technology to solve societal problems in healthcare and nutrition security. We are a program-driven organization. That's our unique nature, where we have stakeholders on board from the start. And we work in a collaborative model. Everything that we do is a collaborative, because then only we can think of delivering something to the society. And I'm using this slide. This slide is not specifically made for this, but this generally conveys what TIGS does and how you can relate with your activities along with ours. So we have several three main research programs: infectious disease, rare genetic disorders, and crop improvement. And these are our core research areas. And to go with that, we have tech implementation platforms. We have a tech implementation team that whatever assays are developed, we see that they reach people in means of policy or means of product. And we have community engagement and policy advocacy, because many times when you come with solution, if it is very high-end science, we may not have the policy or regulatory system, and therefore you need to design that. So you don't have to read it, just to give you an impression that our crop improvement program which is less relevant here, but it's in a larger sense it's relevant, because we need food no matter what other things we do. So we work on how to make a climate resilient crops which requires less water, how to have crops which are more nutritional. So there is a huge, we are sufficient in food production, but we are deficient in nutrition, and how to make our crops more nutritious, and how to use eco-friendly and sustainable methods of pest control, because that also is part of nutrition, because we end up eating a lot of toxic chemicals which are used for the crop protection. So it becomes a counterproductive. And so our model is to how we define our success, how we do what motivates us. One is, what difference does it make to the society of what we are doing? So that's how we count whether we are successful or not. Are we changing the way things are done, things get done? If our research is not contributing to that, that means we are not contributing much. And are our solutions that are economically viable and socially acceptable? If you make something which cannot be used by people, particularly out of pocket, then that means we are not reaching what we should be doing. So as I said, we use a collaborative model, and we work in partnerships. We actually focus on this strategy. So first of all, we are already in a campus called Bangalore Life Science Cluster, where we have INSTEM, NCBS, CCamp, and we are all in a large University of Agriculture Sciences campus, which is not only nice campus, but also has expertise of very, very unique kind, and we get benefited from that. We have lots of collaboration with research institutions, with NGOs, with industry partners, with government agencies, and hospitals and clinicians, because that is how we reach to our stakeholders. Otherwise, we are very small institutions. And our aim is, I'm just putting this that you can imagine your research problem or your clinical problem and put in this pipeline, that we develop something in the lab which involves a scientist. Then we take this solution that we find in the lab to tech implementation team that will interact with the industry partners, and through industry we go with the end users, and that is how we take the solution that reach society. So I'm very happy to work with clinicians who always work with the patient, because we cannot touch patient, and this is how we can reach and solve the problems. This is again just to show that by doing this in very short period of time, we are able to deliver several solutions already who are that are being used in the not exactly for diagnostic but for research use and for surveillance purposes. Our industry is already making them at commercial scale and distributing. And we are a small institution, a very young institution, but we have grown in past few years from a very small number to 150, which is decent number. We have PhD program, we can think of collaboration in those, and we are supported by 120 technical support teams that help 20 scientists in doing what they are delivering. So thank you very much. This was a brief introduction of what TICS is capable of doing, what we intend to do, and we can deliver only when we work with the institutions like you. We have made large contributions at national scale, even being a small institution, in things like environmental surveillance and developing policies for environmental surveillance, implementing it in large scale. So we work most of the things that we are doing, we work with the government agencies, because that is how you reach society. So lots of our programs are supported, not only financially supported, but also collaboration with the organizations like ICMR and DBT. So thank you very much, and look forward to discussion. This was just to show an initiate discussion which we can take further during the day. Thank you again.
All of us come from research organizations, which is, where there is an infrastructure well developed. And you will get a tour of this infrastructure here, what kind of infrastructure they have. But they are also now setting up a very large research labs. Currently, there are a variety of functionality of a hospital and medical college. They have fantastic infrastructure. They already have some research labs, but they're going to expand and they're going to make it much more modern. So they're going to be about 50,000 square feet, you said, you're dedicated for research. It is almost the same as, maybe large institutions, maybe more than yours, almost same as CCMB maybe. Usable area of 50,000 is large. With all the other infrastructure, is already support system is available, like this kind of a seminar hall. Only dedicated for research 50,000 is going to be a large, right? Ulas? Yeah. So in that context, I request all others who speak also mention about what is that we could do together. Who is next?
Can we? Thank you so much, sir. At 11 o'clock.
So very good morning to all of you. I must thank Shashi and team from Subbaiah Institute of Medical Sciences for organizing this very essential and important meeting, because every medical college should have some kind of research. Therefore, the benefit would go to a common people. So in this talk, it's kind of mixed information I'm going to provide. Some basic, because some students are here, and some little advanced one. So as a geneticist, I strongly believe that everything is depending upon our genetic makeup. So whatever happens, either we get the disease or get therapeutic response or no response or adverse response for a particular drug, or we get infection, no infection, everything is depending upon the genetic makeup which we have. Already genetics being used in the clinical setup. I'm sure that in the coming year, at least every single individual who's born is going to get benefit out of genetics. Interestingly, just want to give you what Francis Collin, who was involved in human genome sequence, he said about genomics. He said, genomics is a shop manual with an incredibly detailed blueprint of building every human cell. It is a history book, a narrative of the journey of our species through time, which generally we apply genetics to understand this. And for this meeting, it's very important to know that genetics or genomics is a transformative textbook of medicine with insight that will give healthcare providers new powers to treat, prevent, and cure the disease. So, what we can broadly gain from genomics? We can identify a novel deleterious mutation which can cause the disease or associated with any form of medical importance. And once we identify the mutation which can cause the disease or associated with any other medical form, so one can design experiment for better or improved diagnosis of a disease. So this also can provide additional information that to give a counseling for people who are not well aware of the genetics, that how one can prevent at the level of getting the disease or getting the baby with the disease and so on. So therefore that is very important. And one can also develop a personalized medicine, or one can avoid there is certain mutation which is not going to give any therapeutic response. So one can use alternative drug and so on. Of course, one can also understand the genetic basis of infection and infectious disease, which I'll give some of the examples in the coming slides. Of course, can be also used for medical legal applications. I just want to give you one of the very famous example of, we know that BRCA1 can cause the cancer, breast cancer. There's a 87% of risk if somebody has BRCA1 mutation for breast cancer and 50% for ovarian cancer. And knowing that Angelina Jolie's mother suffered with the cancer, and she has been diagnosed with the same mutation. She doesn't want to undergo that kind of suffer. So therefore, she decided to double mastectomy. And you can also look at the publication which came after that. Somebody systematically looked at it, and this is the average of the people who have done the double mastectomy. But after Angelina Jolie has done that, and you can see it has gone from 20 or 22% to 70% of the people who started, of course, slightly going down the subsequent year. So that means even for the diagnostic, somebody who is very famous, if he or she does that, and the community also try to follow that, and this one also has to think of that. And the medicine, common medicine which we use, not all the medicine or everybody is getting benefit out of that. That's because of the mutation which we have. The genes that metabolize those drugs determines how much of dose is required, how much of whether the person will get therapeutic response, no response, or adverse effect. That's even more very important. For example, these are the things which are commonly used drugs like antidepressant, asthma, and you can see that how many percentage of people were not really getting benefit out of those drugs. And precisely to look at one of the commonly used drug, warfarin, and this drug has been standardized starting with low dose and being optimized. This is one of the studies which was published in New England Journal of Medicine some time ago. The authors actually looked at the record and sequenced the gene, and they found that gene called vitamin K epoxidase complex 1 is a target gene for warfarin metabolism. And they have sequenced the people who have been treated, have been optimized, and took all the record and found, and based on the mutation, they categorized people into 9 haplotype, because these are some of the mutation which is existing in the full gene, and putting together and found that individual with this particular haplotype H1 have been optimized with 2.9 milligram of warfarin per day. On the other hand, look at H7, there are few mutations. In place of C, there is a T. Similarly, there are very few mutations. So that few mutation determines that the individuals who are carrying those mutations require double the dose of warfarin, because this has been optimized earlier. And this is a study which was done much later. So if somebody knows this from the beginning, they don't have to wait for a long time to see whether they are getting benefit out of that or not. Straight away, if somebody has haplotype H1, then they can use 3 milligram of warfarin, or instead of that, they have H7, they can directly go to 6 milligram, double the dose of warfarin per day. And few examples from the infectious disease. This is one of our own study where we looked at the manners binding lectin gene, and you can see actually the frequency of low MBL2 haplotype is significantly higher with the severe malaria, this one. You can see in the mild malaria is less, and this is asymptomatic. Okay, so this determines that manners binding lectin level is important to determine the infection of a disease. This is in case of malaria. This is in case of the rheumatic heart disease. You all know that rheumatic heart disease is progressed after the infection, and there are multiple ways it goes, and one way is rheumatic heart disease. There are several mutations has been identified, and this is systematically classified, the mutation which is in the poly-A site, and you can see instead of T, if there's a instead of A there is a T, then you can see that significantly increase the susceptibility to rheumatic heart disease. That mutation you can see reduce the MBL level, therefore they are having more possibilities of having rheumatic heart disease. These are some of the very, very unique mutation present in India. And there are several other such studies, again I am getting from my own study, that the cardiomyopathy, there are 3 different kinds of cardiomyopathy, you all know, dilated, hypertrophy and restrictive cardiomyopathy. So one of the genes responsible for causing the disease is myosin binding protein C3. This is a 25 base per deletion, again precisely present only in India. Then we extended the study to look at its distribution all over the world. We found interestingly that is present only in South Asian population but not in other countries. That means this mutation we estimated must have originated in India about 33,000 years back, and there is a lot more background which I don't have time to tell all of those, and this is restricted only in India. That means this mutation is very old, so because of population admixture it got well spread, right? When it is the heterozygous condition, it causes the disease only after age of 45 or 50. But in the homozygous condition, that means both the copies of the chromosome carry the mutation, the individual get the disease and die at very, very early age. So this is the one which is well spread across India and also South Asia. On the other hand, there are certain mutation which causes a disease, particularly if you look at this disease, junctional Herlitz epidermolysis bullosa, which is caused by a mutation 11 base per deletion in LAMB3, exclusively present in only one population with very high frequency, the population near Madurai in Tamil Nadu. We screened all over India, there was no mutation. We screened 16 lakh individual sequence which is in the database, but none of them carry this mutation. That means it's exclusively present in India. So why such heterogeneity exists in India, right? Already Shashi pointed out that we have very diverse population. As you see here, we have population typically look like Africans, we have population typically look like European, we also population typically look like Southeast Asian, but there are populations which are very unique to India. So that we have been studying for the last 3 decades. I'm not going to detail of that. But if you look at precisely what is the reason for this, and based on our own study, you can say that the very first out of Africa human, you all know the human population arose in Africa. The very first population who migrated out of Africa is in Indian subcontinent, who are none other than the Andaman tribes. I don't know how many of you are aware, this population typically look like Africans in Andaman Island. Subsequently, there are several migration until very recently, like Siddhis, Jewish, and many more population who have migrated in the recent past. And we also have a population who are geographically isolated in many places, who are in isolation. And most importantly, Indian population practice endogamy, means marrying within the community. We predicted that at least for the last 2000 years, every single population is practicing endogamy. Therefore, whatever the mutation which happened in that particular population remains there. If the population gained a mutation which leads to disease, is going to be restricted only there. That's why there are large number of population specific disease that are existing in India, which we looked at population from South Asia and found there are large number of populations who are having scientifically or technically you can say IBD score, identity by descent. That means if you take 100 individual from the same population, most of them will have the same haplotype, means same sequence. That means they all come from a common founder. Therefore, we predicted that at least 1/3 of the Indian populations are expected to have a population-specific recessive mutation that might lead to population-specific recessive disease. You can see that these two are Ashkenazi, Jewish and Finnish. With the tiny IBD itself, there are 25 to 30 diseases have been already mapped. But you can see the consequences of large number of population who have multifold higher IBD than the Ashkenazi, Jewish and Finnish population. So there are large number of genetic or genomic studies are being carried out. At least I'll give you a couple of the studies which have been sponsored by DBT. Fortunately, I've been involved in those studies. One is the Genome India study. I think Shashi already pointed out that there are 10,000 genome has been recently sequenced, and they're all has been deposited in IBDC, which will be soon available for anybody to make use of that. I'll give you a couple of examples how we gained, how one can make use of this for the better benefit of common people. This is one. Second one is pediatric rare disease program, has been taken lead by CDFD, where Professor Ullas is heading that institute, which has been launched some time ago. One of the examples which I wanted to tell you about these studies, you all know clopidogrel is being extensively used for cardiac problem and stroke. And the mutations in the CYP2C19 determines the dosage of clopidogrel. Based on the mutation, there are 3 categories has been classified. One is intermediate metabolizers. These are normal metabolizers. These are poor metabolizers. Right? I think color is not that distinct, but this is orange and this is maroon color. Right? But when we looked at this 10,000 genome, and we found that approximately 15% of them are poor metabolizers, that's what given here, and 45% are intermediate metabolizer. That means approximately 60% of the people who are taking clopidogrel are nearly not really getting any therapeutic response, right? But on the other hand, if you look at the data which we took from RAG, that about 10 crore Indians, they are taking clopidogrel every single day. And the economic problem is approximately 185 crore spent annually for this particular medicine. Then you see that 40% of the people only get benefit out of this, 60% are not really getting benefit. Therefore, one has to come up with a new drug for better treatment. This is another example. There's a gene called BCHE that is essential for metabolizing succinylcholine. You all know that's given at the time of surgery along with anesthesia. A specific mutation, this actually lead to the individual who have been given the succinylcholine at the time of surgery along with anesthesia. They don't actually metabolize the drug, but they will be remain with the anesthetic effect for very long time. Sometimes it leads to paralysis, sometimes it's also life-threatening. When we looked at, because this mutation was found only in a community called Vaishya, okay, or in Andhra Pradesh or Telangana. There are also small group in Coimbatore and Tamil Nadu. No other community has been reported with this mutation. When we looked at this 10,000 individuals, we found a total of 44 individuals from 29 community are carrying this mutation in heterozygous condition. Because when it is homozygous, only then it leads to that kind of problem. Then you can imagine most of the populations are practicing endogamy, and maybe after several years or several generations, because of endogamy, the mutation load in the population is going to increase, and there's a possibility of having homozygous mutation. Therefore, they also can face similar problem of what Vaishyas are facing today. Right? So therefore, there could be, there's a public health policy needs to be come up with to screen for such mutation. This is just one example. There could be many more to for all those who are going for a major surgery. This is another example that it came from pediatric rare disease. It's a mutation in GJB2, which leads to hearing loss. Again, it was not well known, that has been distributed all over India, but we found that this is found in 47 community, and some of them have very high frequency also. So this is really coming very, very interesting. This is another example which came from pediatric rare disease, a gene called SRD5A. I think somebody is working here on DSS, which came from the previous presentation. When this mutation is in a homozygous condition, in XY karyotype, it leads to ambiguous genitalia and other DSS features. This is not well known to be associated in large number of population, but we found some of the population, 9 population, at least 1 each and 3, but interestingly, one population from Odisha, one of the tribal community, have 11 individuals with heterozygous and 2 with homozygous. In a XY karyotype, they will be actually with DSS, with ambiguous genitalia. But when I asked the people who collected the sample, go back and found that these 2 are female. In fact, they are mother of the kids. That means in the XX condition not affecting, something like the androgen receptor gene mutation in XX condition, not going to lead anything. They are fertile, but it pairs that abnormal chromosome with androgen mutation, that's X chromosome, which pairs with Y, then it leads to XY sex reversal phenotype. So these are some of the information which we are gaining from both the big genomic work that it leads to requiring more of public health policy for screening that. Okay, so now I'm coming what we can do together. I have interesting agenda that I know that this area is known for anti-kudu disease, right? We initially, with the help of Mushtaq, who is from Mangalore University, we collected few hundred samples and we did exome sequencing. Really, we are not getting the real signal which may be responsible for the disease. But now we wanted to go for the whole genome sequencing with long read along with epigenetic information. Therefore, we may find something interesting. And you are being there in this area, we don't really have any clinical information or other biochemical X-ray or MRI information. So this will be a good point for us to do some of the clinical examination. These are a lot of biochemical tests which we just listed out from other studies with the bone abnormality, not precisely the hand-kudu disease. So all these can be done with radiological examination. So therefore, we can address this issue of hand-kudu disease. In addition to that, we have been in CCMB using genomic tool. My own group is studying the population studies to understand population origin, migration, and so on, and also using that for understanding some of the diseases, including mitochondrial disorders, group of mitochondrial disorders which affects almost all the organs, including eye. I have seen this chronic progressive external ophthalmoplegia also has a lot of impact on eye. Cardiovascular diseases, one I mentioned cardiomyopathy, rheumatic heart diseases, and other diseases. Some other rare diseases which I have shown, and some diseases we are in collaboration with Rakesh and Tix, we are taking it to the society. Therefore, the society can benefit out of that. Male infertility, DSS, and a few more. In fact, our population genetics study helps for better understanding of these diseases. So I'm sure that this networking program throughout day which we are going to have will help us for understanding each other's capacity and expertise, and probably can come up with some kind of collaborative projects. So with that, thank you all for attention.
Yeah, so again, it's been a pleasure coming to Subbaiah Medical College, and it's really nice to see that, of course, it's very difficult to say no to Shashi. And Shashi is one of the most well-networked people in India and elsewhere. It's also nice to see that there are more number of people now in the community who are willing to look at science and impact beyond publications. And I think it's just the right opportune time for all of us to come together and probably do something more meaningful. So all that I'm going to do today is kind of give an overview about the kind of work that the organizations that I'm associated with are doing, but it's not necessarily showcasing the science per se, but telling you about how one can possibly shape research initiatives, specifically in the context of clinical and public health work, which I think is of relevance for you. So I've put this very provocatively, said that wake up, smell the looming disaster and join hands. I have multiple loyalties, and so of course I represent all of these organizations. I'll start with some, very politically rhetoric kind of statements, but these have impact for all of us and particularly for youngsters as well. Somebody whom I value a lot, Dr. Anil Kakodkar, who is the ex-DA secretary, often keeps saying that it's institutions build nations. And I say people build institutions. And for example, Subbaiah Medical College is one such example. And I think there are people here sitting in the, all the experts who actually have built institutions. And then the question is, how do we contribute and how do we come together and bring institutions together to make things happen? But before I even tell you about what the organizations that I am directly associated with do, I'll give you a story about another similar medical institution in the country, which largely works on rural health, like pretty much what Subbaiah Hospital does. And now we are intimately associated with them, and of course Shashi also knows them. This is BKLW Rural Health Hospital, which is Valavalkar Hospital, which is present in Chiplun district in Maharashtra at a place called Darwan. Not so long time ago, Darwan was a sleepy village, and basically it looked like pretty much any other rural setting. A very motivated trust and a very dedicated, husband and wife couple who were doctors decided to make it their home, and then now today it's one of the most vibrant medical research initiatives or institutions in the country set in a rural backdrop. They have schools, they have nursing colleges, they have fantastic clinical care facilities, but also a lot of social outreach activities. In fact, I've been overwhelmed with their community health impact. Now, we got to know them, again, thanks to Dr. Anil Kakodkar and also Dr. Ramaswamy. I think many of us know these individuals who happen to be on the board of their advisory council. And then these two people knew me and the organizations that we were working with. They said, why didn't you come together and help them to set up something which is exciting? So one of the things that I would like to highlight, how Darwan Hospital over about 3 decades has made a significant impact in terms of healthcare in rural India by looking at everything that happens from reproductive or adolescence age group all the way through gestation, neonatal health, adult health, and even beyond, looking at the entire life cycle of an individual. And they've intricately coupled with social interventions. And in fact, they do a lot of work for handicrafts, a lot of work on sustainable agriculture, in a sense that have brought the community together. So, and this is important for even a medical college like Subbaiah Hospital, because if you want to gain the trust of a community, it's not just sufficient to do only medical thing. You need to be connected with the community. And again, most of the institutions that we work with have this strong community presence beyond the hospital setting. And I think that's an important take home, because otherwise the interventions you will come up will not have an impact. Now I'll come back to Darwan Hospital later, because they're now working with us very closely. But I'll start with another major problem. Let's move away from rural setting, but also go into more urban settings. Even here, there is a large disconnect between what happens in the lab and what happens at the clinics. There is a translational gap. But there's a big gap between medicine and medtech. And even if you understand what happens at cellular or molecular levels or at a clinical level, it's very difficult to translate them into community health settings. I'll give an example, which is a real life example, that we work with several communities and several networks in the world. And I was sitting in a International Atomic Energy Agency, which is the IEA, which is a UN body network meeting. A public health expert from Kenya told me that all the micronutrient supplements and iron supplements they were giving had no impact over 10 years in the community. And they were surprised that despite having high compliance in terms of taking the thing, they all used to come diligently take the boxes and go home. And then they realized that all these macronutrient supplements, the community was feeding them to the cattle, okay, not to the children. It sounds funny to us, but for them, making that extra money was more important than worrying about stunting or wasting in children. So the reason I'm saying this is, again, community presence becomes extremely relevant even for institutions like Subbaiah Hospital, and that's something that we need to worry about. And what you think will work in a population may not work in another population, and nothing to do with biological causes, nothing to do with genetic causes. It would be purely social acceptance of an intervention that you're going to come up with. So that's important for us to think about. Now, what is really killing us? There are many diseases. And of course, one of the biggest things that, I'm taking the leap forward from what, segue from what Tangaraj said. Genetic diseases are one of the biggest causes of everything that we think affects human beings, particularly pediatric rare genetic diseases, which are underdiagnosed, underrepresented, and understudied. The second thing is, of course, non-communicable diseases, and the landscape of diseases has changed over the last 30, 40 years, and especially post-industrialization, but more recently because of sedentary lifestyles. And it's very well known that currently the mortality associated with non-communicable diseases is about 50 to 60%. In fact, non-communicable diseases are killing more than most other diseases, and this is only increasing rapidly, even in rural India. In fact, again, the data that we know from Derwent, actually there is high incidence of dyslipidemia, high incidence of cardiovascular diseases, even in rural sectors, and these are not anymore urban diseases which are associated with the rich people. What is also worrying is the fact that the age of onset of diseases is accelerating, and in fact more younger and younger people are getting affected by these diseases. Now this is more for people like Shashi and Rakesh and Tangaraj, who sit in the DBT committees and all such committees who decide money. Unfortunately, the amount of money that goes into non-communicable diseases research is almost 10% of all the health research that we fund, although about 50 to 60% of that happens because of non-communicable diseases currently. And this is probably worse in India. And most of the work, of course, goes for HIV and AIDS. I'm not debating why you should not spend money there, but there's a huge funding gap in terms of what is required to tackle this. Now, I'll briefly give you overview of CDFD, because that's the organization that I'm currently leading administratively. And I'm also using this as an opportunity to impress upon you the importance of genomics and how Subbaiah Medical College or other such regional medical institutions should leverage and connect with centers of excellence or facilities that are available in many of the other premier research institutions to not only help those institutions by providing unique cohorts, but also gain from these institutions by accessing technologies. So CDFT is a unique organization because we do a lot of basic research. In fact, we are supposed to be one of the best DBT institutions right now. And we do globally competitive research. We publish in some of the top journals in biology that all of us can dream about. But we also have a service associated with it. One is, of course, diagnostics. And Tangaraj, when he was associated with CDFT, also initiated this rare genetic disease program. Now, forensics and diagnostics is one of the mandate for CDFD, and we have been doing this extremely well over the last 4 or 5 decades. And in fact, again, it's in the mandate, and we are now the leading organization to provide cutting-edge tools, but also develop new methods that are important. We are probably right now having the best genomics facility in the country with what is called NovaSeq X Plus, which can do up to 64 genomes at one go, and we are offering these services at very low cost for institutions which are doing public health intervention and actually at the societal level. Now, I would like to invite you also to think about connecting with our rare genetic disease program. This is largely led by Ashwin, who is himself a pediatrician and actually practices, and he also does clinics in Goa. And CDFT rare genetic disease program is actually now connected to several rural and urban hospitals all over the country. And a recent DBT push through this, thanks to our efforts, has also led us to become the nodal organization between both DBT and the Health Ministry to provide this kind of research environment and service for all 100 aspirational districts in the country, which are called Ummeed or Nidan centers. And now we are scaling much more, and we are providing a lot more access to technology and medical health. And the reason why you should belong to these such networks is that not only do you get access to technology, but you also have a lot of peer learning that happens from other similar research centers, both in urban and rural settings, and there's a lot of cross-pollination of ideas that can happen. Now I'll go from here, from genetic diseases to non-communicable diseases, because that's not only my area of research, but that's something that we have initiated in TIFR, and I'm going to talk to you about this initiative. And again, the purpose here is not to tell you what you should do, but rather to say that you have to seek opportunities in what relevant problems exist in your population, in your settings, and use those opportunities to connect with the networks and think about how you can do end-to-end, really high-quality research, which leads to discoveries, but also leads to translational impact. Now, this photograph I put is not to show that I was also young at some time. This is just to acknowledge my other co-conspirator, Mahendra Sonavane, with whom we do this. And Arumda is based on a simple and yet, I believe, a powerful mission and vision. We are not a simple research organization or a unit. Our mission is to empower researchers, technologies, and policymakers and community health partners. And I'm going to come back to this empower word, because we are not just doing cohorts, we are not just doing research, we are actually truly empowering community health partners. And I would like to emphasize community health partners because you are a community health partner. So we have a physical center which is largely present in TIFR Hyderabad. We have virtual network of institutions. I'm going to tell you that, and we'll be very happy to associate and help Subbaiah. We are now on scale the largest consortium or a unit or a research organization that has brought together basic biologists and clinicians on this scale. So what is the overview and mission? And I think I've said this earlier as well. We all know that all diseases run into old age. Even the risk of infectious diseases increase when we age. The outcome of infectious diseases is also based on the non-communicable diseases, very evident from COVID. People with comorbidities were the ones who suffered the most. Outcome of infections, both peripheral and systemic, is known to be differentially affecting healthy individuals and people with obesity and diabetes. All of us know this. So given that fact that all diseases run into old age, there is no dedicated organization to look at metabolic disease and aging in the country. And in this context, we began to work and put up this particular center called Arumda. We now work with about 40 organizations, both in the country and elsewhere. And of these, I would like to specifically highlight 3 or 4, which are very vital partners for us. One is CMC Vellore and the entire All India Institute of Medical Sciences network. We work with 3 major aims, but also associate with other aims. And the other major partner here is the International Atomic Energy Agency, which is the IAEA. And of course, we are now forging a lot more ties with organizations like UNICEF, the Gates Foundation, and the Wellcome Trust. Now, of all the problems that we could have picked up, we thought that we should start with something which is very fundamental and has impact across lifespan, and that's called double burden of malnutrition. I think many of you know what double burden of malnutrition is. It can be defined in multiple ways. At an individual level, double burden means that you can be micronutrient deficient but also have obesity. At an individual level, you pay the price when you're young because there is childhood stunting and wasting, but also you have higher risk of developing cardiovascular disease, obesity, diabetes, and even cancer later in life. At a household and population level, it can be described as coexistence of both obesity and stunting and wasting within the same populations. But what is worrying is not the double burden of malnutrition. What is worrying is what is called triple burden of malnutrition, which is typically called as hidden hunger, which is the deficiencies in the nutrition that we consume in the form of diet. But much worse is the inability of the nutrients to get absorbed and utilized in the body, which is called resistance to uptake and utilization. And we don't know why there is resistance and to uptake and utilize these nutrients. For example, iron supplements have been given for a long time. Of course, there's a compliance problem, but even if you give iron, you are not able to solve anemia beyond a particular tipping point, because it looks like there is resistance to uptake and absorb iron. And all of these have direct implications on child and growth, and of course at maternal health as well. Now, why is this relevant? And this is beyond only Arumda, and this is where all of the partner organizations should come together. The median age of Indian population today is about 20 to 25 years of age. And by 2050, which is also the Amrit Kal Bharat that the Prime Minister has envisioned, the age will be around 50 years, and which is where all typically diabetes and obesity or cardiovascular diseases set foot. Now, if half of our population or 1/3 of the population is malnourished, this population will have a higher risk of developing non-communicable diseases later in life. That means you're likely staring at pandemic proportions of CVDs and obesity and diabetes by 2050 within the population. It also has huge economic cost. The estimated cost of childhood malnutrition is around $30 to $40 billion in 2030, but the estimated cost of early onset NCDs is supposed to be much higher than this. Now, if you're not convinced by this, there's this concept called DALI, which I think many of you are familiar, which is called disability adjusted life years, which is a combination of both the number of years you lose because of early mortality and the number of years you lose because of morbidity that you live with. In fact, child and maternal malnutrition, higher dyslipidemia, dysglycemia, all of these together combined give you this impact. And until recently, there's this general assumption that most of these diseases affect only the urban population and very little is affected in the rural, and we assume that rural population usually live a more healthier life. But unfortunately, our own data and the data that is emerging from surveys from NIN and ICMR shows that rural population is actually suffering from the same kind of diseases pretty much with similar prevalence levels. Now, let's see why this is also important for multiple organizations to come together. Even if you think about a particular disease that has a set of symptoms, now these symptoms could have emerged at any point in life. That means there is a life history or a trajectory that has happened. It could be intergenerational, this could be gestational, neonatal, childhood, or any other period in life. But it also has multiple biological causes, and it's important to understand all of these biological causes collectively. So I'm going to skip a lot of details and tell you what cohorts we are right now doing, and we are increasing our cohorts as we go along. And one of the first things that we said is we'll define what is supposed to be a healthy adult cohort, which is not really available. And this is something that is being done with AIMS and CMC Vellore, with some of the top leading endocrinologists in the country. We said we'll take absolutely healthy individuals between 25 to 45 years of age. That means there is also not confounding effect of an age-associated decline in physiological fitness. Rigidly screened for absence of comorbidities, rigidly screened for prediabetes, and it is here itself, it's shocking that to get 80 healthy individuals, we have to screen close to 300 individuals. That means, see already the tipping point, that is, all of these individuals are at risk of becoming, compromised later in life, and we are talking about 25 to 45 years of age. Now, we are doing this across 3 populations, and we are now increasing it to 2 more populations, which is about 5 populations. The second thing that we are doing is addressing malnutrition in children, which is also a concern for most of the populations. And malnutrition is not only undernutrition. Malnutrition is both overnutrition and undernutrition, rather imbalanced nutrition. The third most important thing that we are doing, I'm going to highlight that, is normative baseline for childhood development. But before that, something that might be of interest for Subbaiah Medical College is also adult girl micronutrient deficiency program. Now, unfortunately, girls are generally ignored in the healthcare system, adolescent girls even more. And we don't understand the scale and scope of micronutrient deficiencies that are affecting this particular vulnerable population. Even in the urban settings, it's ignored, but in rural settings, it seems to be much worse. Now, we are doing a large study. This is about $5 million grant from Gates Foundation, where we are looking at the scale and scope of adult micronutrient deficiencies in adolescent girls, And then we are trying to see what kind of interventions can help them. But rather than simply looking at in the levels of micronutrients, we are associating these with clinical outcomes. In fact, we are doing very deep dive neuroendocrine pathways, deep dive metabolomics, and several others. We can discuss the details later. Right now, we are spread out across the country. Together, we are now doing in parallel about 12 cohorts. Of course, when I say 12 cohorts, it combines both the number of sites and number of studies. We work with some of the best organizations that you can think about, like I said. And the reason I said best is not simply by name, but best because what Shashi said is the rigor of quality of data that they collect and the community reach that they have. And I really want Subbaiah Medical College to actually have that rigor and ethical practices that are essential for, if you have weak epidemiological data, no biologist will be able to give you any answer. The fundamental principle of anything that you want to do at a public health level begins with collecting really rigorous, stringent epidemiological information. If you don't have good epidemiology department, you can't do much, because then you can't have a public health impact. So that's my learning from working with a lot of these centers that we have learned with. And all of these centers have actually invested in public health and community work. Now, this is just an example of what I said we can do is, for example, with the malnutrition cohort, we are doing a lot of work and looking at when does double burden of malnutrition set in children. And surprisingly and shockingly, in populations which like CMC Vellore, which is actually peri-urban, in fact, it's a very modern lifestyle as well, the double burden seems to be setting at the age of 6 years, which is shocking. You have very high risk, prevalence of both stunting and wasting in these children, but also a high risk of obesity, which seems to set in very early at the age of 6. Now, that means this gives you an indication of when you want to intervene in terms of taking care of the child health. Now, we realize that just doing research by ourselves is not good enough. Like I said, Arumda is mission to provide empowering support to communities. And in this context, we set out to establish the first National Analytical Facility for Nutrition and Metabolism Research, and also something called a Simians platform. Now, why is this important? Because all of us realize that ultimately, whether you have a disease because of a genetic cause or an environmental cause or an epigenetic cause, ultimately what matters for physiology is the metabolic fitness. And unless you understand metabolic fitness, you will not be able to do much in terms of treating the disease. So there was a dire need for having a centralized facility which would benchmark to the best in the world and also give access to everybody in the country. And this is not simply serving every institution in the country, NCBS, CCMB, CDFD, TIFR, all of us have mass specs. But never have such facilities been offered on a platter. Of course, we are all supposed to give service. We do give service, but not as a dedicated service mode, in a service mode. So we set up this National Analytical Facility for Nutrition and Metabolism Research. The second thing is, I'll highlight one important thing. I might look lean, but you ask a doctor, he'll say, yeah, you're fit because your BMI is low. But actually, BMI being low is not good enough, because you might be physiologically obese, because this concept of discordant fitness that is measured through BMI and body composition. Body composition gives you the lean mass to fat mass ratio that you'll never be able to get from a BMI. How do you measure body composition? Typically, you do DEXA, right? Because that's what doctors do, or a BIA, which is a bioimpedance measurement. Bioimpedance measurement has a lot of error. DEXA is, of course, exposure to X-ray, expensive, only present in tertiary healthcare centers. Now, how do we make sure that these things are available in rural healthcare centers with every ASHA or an Anganwadi worker? So, about 6 years ago, before we started Arumda, we didn't even know this. Apparently,
for giving me this opportunity to come down here. So I'll just briefly talk about. So I work at the Translational Health Science Technology Institute. So where we don't have clinical setting, but we are exactly, you can say, collaborating with a lot of clinical hospitals in and around. And now we are here so that we could go into external validation for some of the markers that have been identified. So coming to the basis, I would just like to see how, what you can say, define my experience that how we set up a cohort over there and how it can be replicated here so that we can think about collaborations in that way. So Ullas gave a very brief about how it is very important that if you really need to go into the biological disease, you also need to know that you have to risk stratify the population that you're working with. And for that you need a lot of epidemiology data so that you know you're working with the right people. So this is a Garbhini cohort, which is Interdisciplinary Group of Advanced Research on Birth Outcomes DBT India initiative. This was funded in 2015, and since 15 the cohort has enrolled 12,000 mothers, and it is a prospective cohort where the data is being taken as an epidemiological data with medical history, clinical, biochemical marker, routine testing. So all that is done at all the 4 or 5 antenatal visits, at delivery, and also we are following the mothers at postpartum. So not only epidemiological data, but we are also collecting biospecimens when they come in contact at the site, because we also do community studies. So we do go to their houses also, but when they come to the site, we collect the samples because it's easiest for us to collect the samples in the hospital. So we are collecting it at less than 20 weeks, which is very important phase of gestation, which is the first trimester. Then we collect it at 26 to 28 weeks. So this was done at Gurgaon Hospital, which is a tertiary care hospital, where they did not have a standard of care for detecting gestational diabetes. So with this cohort, we started the OGTT at the clinical setting. So it's still on now, even after the cohort. So 26 to 28 is for gestational diabetes, so we collect our samples there too. Then we have ultrasound of 30 to 32. This is very important for stunting. So you need to have imaging markers also. So that is why we continued the ultrasound at 32 and also 35 to 37 weeks. Then we collect samples not only of the mother, but also the breast milk and the baby, infant samples. For any metabolic diseases that could happen, because we are going to follow these babies for another 2 years. Then we collect a postpartum sample. So this is all I just wanted to talk about, that how beautifully, it's not one person who can work, silos that can work. So it's a whole team which is working. Regional Center for Biotechnology, THSTI, then we have Safdarjung, AIMS, New Delhi, and also NIBMG that are working in this cohort. Go ahead. So this is a whole team. I just wanted to put the team in front here, and Dr. Shinjini, the second person out here, is the main person who is leading this cohort. And it was all Dr. Bhan's idea that we need to have this translational institute where we can have the clinicians, the immunologists, the biologists all working together with these networks. Next slide. So now to do anything, as Ullas mentioned, we need to clinically verify that the data is correct. We also need to know that the samples that have been collected, processed, and carefully archived with some enrollment IDs, unique IDs, are kept in a biorepository which follows best practices. So the first thing that we did was, the first 5 years of the cohort went into setting up the community cohort, setting up the hospital-based cohort, and also at the institutional level, setting up the biorepository, which has turned out to be the first internationally accredited ISO 20387 biorepository of DBT. So we have 1.6 million samples that are beautifully coded, individually coded, and they are unique IDs. We have also time-stamped them, not from the collection to the processing, but also the archival and the retrieval of the samples. So this is what we have done, and because we had a biorepository, it's not only that we share the samples amongst the PIs, now we have the embargo period which has opened. We share the samples, we have made a governance process, and DBT, the Department of Biotechnology themselves, have nominated some of their members for an access control committee for this biorepository to be a national biorepository now. Interestingly, from the biorepository, we also teach, you can say, or empower the other institutes who would like to follow the same principles and have their quality management systems as robust as ours. So we give workshops. We have Gunvakta Yatras that we do with Quality Control India, that is the Quality Control Council of India, and NABL. So apart from that, we also have trainings of biobank and also help them with the study design consultations, that exactly for what type of disease do you need to take the samples, you should have these processes. And from this biorepository, all the institutes that were collaborating with Garbhini have come up with some markers. Some have come up with panels which are SNP panels, genome panels, some have come up with microbiome panels, the others have come with proteometabolic panels, and it is all because it was a concerted effect not only of the clinicians but the data management people, the technicians who put up this biorepository, had their data ready and the clinical data ready to start with the sample. So just 2 more slides, and this is what I am interested in. So after the biorepository, I started with my work, which is the lab of perinatal research. So we work on the placenta, and as mentioned again by Ullas, we work together, but I don't think we collaborate. So the point is that he said that there is a lot of wasting, malnutrition. It's not because the mothers are not giving everything to the cattle, but because the placenta also sometimes cannot transfer it to the fetus. And that is why you have this undernutrition. So that is what we are interested in. And we cannot sample the placenta in the pregnancy. So we have developed a technology of isolating exosomes, which are small bilipid cell extracts which come out into the mother's circulation, and we can take that from the mother's circulation or sera and plasma and look into the cargo whether it is different from a mother who delivered a term baby and a mother who delivered the preterm baby, because placenta talks to the mother, and this is the message we want to understand and decode. Second is we also go ahead with placental histopathology, because I think in your field that is the thing, that is a hard, you say, what do you call it in your hard outcome that you would like to see whether if something has gone wrong. The third is we also are involved in sera and placenta tissue omics, majorly metabolomics. And then we also have started making placenta on a chip, which is a placental organoid, where you can study the genes that you have identified, whether they are affecting the cellular processes, basically the angiogenesis, the development, the proliferation in the placenta. So it's a whole complete thing. And I'll quickly go through those slides. We can skip that. I just talked about the placental exosomes, and this is how they look. So you need specific instruments to look into that you are indeed working with these exosomes. Then you can characterize them at the protein level also. Now this is where I would like to bring your attention to. So we are using an integrated approach to develop screening and diagnostic tools for APOs, adverse pregnancy outcomes. It could be pregnancy complications. So in our cohort, we have 13% preterm birth, 4.3% late onset preeclampsia, and 2% stillbirth. So these are the major, and 14% of GDM. So somehow these are the major things that 4 hard outcomes that we are looking into. With the help of clinical risk factors and also ultrasound, that is the hemodynamic flow rates, which we are capturing for the uterine artery and the mean uterine artery umbilical flows and pulsatory index. Second is the biochemical marker for placental insufficiency, PLGF, SFLT, PAP-A, and all the other folate deficiencies can be also picked up. So we are doing that. And also the markers of inflammation, which is CRP, AGP. These are the causal markers, and they could be better predictors of a disease. I'm talking about in terms of pregnancy. And the last is the novel circulatory biomarkers, where we as biologists come in. So try to identify the novel proteins and the metabolites that are associated with the disease and use them into these algorithms which are already there in clinical setting to improve the accuracy to predict the disease and manage it. So these are metabolomic markers, and this is the last slide. This is the current status of the biomarkers that is in my lab, but mind you, there are 5 more groups who are working on protein markers, microbiome markers, epigenomics, and also genome markers, SNP panels. So we are trying to populate everything over here where we can use for prediction of the hard outcomes. So first trimester, second and third. So we have for preterm birth, for LOPE and stillbirth, we have some markers which have been internally validated in the 12,000 cohort, and we would like collaboration so that we can validate them in external cohorts all over India. Thank you so much.
And thank you very much for this opportunity. special thanks to Professor Shashi and Lata ma'am. I don't have any presentation, but I can give some glimpse of studies or work what we are doing. So, my name is Dr. Anand Kaude. I am basically a pediatrician by training, and since last 20 years or so, I am working in the public health. So, I am from the KEIM Hospital Research Center, Pune, their Vadu Rural Health Program, you can say as a Vadu cohort. At Vadu, we have the health and demographic surveillance systems since last 20 years, and we are following the population there since last 20 years. Initially, when we started, it was around 60,000, and now it comes to almost 2 lakhs. So we are collecting the demographic data, we are collecting the health data. And now I am giving you a few examples how we can use these data to have the really policy things or impact in sense. I will quote few examples. So one is about the, I'm not sure many of you have heard this term or not, Indian childhood cirrhosis. So basically this entity was something to do with the use of the copper utensils for the cooking. So it was a pioneering work by the KEIM Hospital, and late our Professor Anand Pandit has done that work. And then we have shown that this is a copper vessel, use of that utensil and making the cooking in that utensils will give the copper in the body, and that destroys the liver. These young babies, they die in the age of 5 years or 6 years, very early cirrhosis, and it is a refractory cirrhosis. So we have done a lot of work starting from the mechanistic thing and then in community, and now this entity is extinct. So this way we can do, this is one example. Another example is what even Dr. Ullas has mentioned, this is about our maternal cohort. So it was started like the diabetes is a urban disease, not rural. And then we started following our rural mothers for the diabetes. It was late, I think, some 2000, something like that, and we are now in the 3rd generation of that. And we have followed the mother over the period. So mothers, they are all biomarkers, diabetic markers, then their babies, then the baby grows, then those babies, they again married, their babies we are following. So we are following now almost 3rd generation. And what Ullah is saying, it's a very surprising fact. So we can see the early markers of diabetes or metabolic syndromes in this as early as 3 years or earlier than that. We followed them 3 years, 6 years, 18 years, long follow. So yes, this is entirely an epigenetic mechanism, and that's support the what we call as a Barker's hypothesis, right? So it's a intrauterine malnutrition of the baby, then once it is delivered, then excessive nutrition or something, then that is it take up the things and development of the metabolic disease. So that is another important thing. So in this kind of community, we can do this kind of studies and generate the evidence. Then 3rd example is a very common example, like our vaccine research. We have been the Center of Excellence by ICMR and Core Vaccine Research Unit, and we were doing the licensure phase 3 trials for the vaccines. And importantly, we know that currently, recently, in last 10-15 years, India has developed indigenous vaccines. So I'm happy to share that out of these vaccines, 6 vaccines in the program, like Rotavac, Rotacil, Pneumocil, cervical vaccine, all this vaccine research has been done at Vadu at our site, and we are instrumental for that. So such kind of impact we can do if we were with the community. Then another example is like now the important is we are doing this community research, and then we are following the cohort, and we found that the chronic obstructive pulmonary disease, and we know that common the exposure to the smoke and these things, vehicular smoke is one of the cause for that. But when we go into the community in the interior, we found that the indoor air pollution is a cause of this COPD. And we are the first to show that this kind exists, and there is some phenotypic difference in COPD because of biomass use and COPD because of vehicular pollution. So these kind of things can be done when we have the community set up like that. Now about the Subbaiah. So I think it's a great vision by Lata ma'am, and you are at a great position to start these things. So you have the will and zeal by the ma'am. We have the department. So for any health research, I think to my mind, we require only three things. We require the lab, we require the pool of scientists, and we require the community. So you have the community, you have the labs, and everything is difficult to arrange, but to my mind, what is most important, difficult thing is to have the develop a pool of scientists, because developing the scientific rigor is almost very important. We all are a clinician, and in future you will go and do the practice. And therefore, less likely you may be involved in the research or not. But please remember that the scientific rigor is not only developing on these things, research thing or own proposal. You have to start asking the research question from the very beginning. Unfortunately, in today's clinical curriculum, there is nothing based on the research. There is nothing in the research, but you can develop. So you are at very good position since these students, they are very naive. You can develop that temperament into that. Just start asking the research, very simple research question. I can quote a very good example. As Pallavi said, Dr. Bhan. So it was like when the first rotavirus vaccine was generated, indigenous rotavac. So it was started late in the, I think, 70s when there was a diarrheal epidemic in the AIMS neonatal unit. And that epidemic happened, and then that sister nurse, what she observed that those babies who have diarrhea during that NICU epidemic, they didn't get the diarrhea significantly, or their diarrhea was very mild over the past. And she has observed that, she tells this observation to Professor Bhan. And then that strain was isolated, 116E. And 116E was taken for vaccine development, and that's how we get the first indigenous rotavirus vaccine, which is completely developed in India, and now it is in program. And now you can see the effects that there is a tremendous reduction in the rotavirus diarrheal disease. So a very small observation as a clinician, as a medical professional you can have, but the impact can be there. So please keep that mind open and start asking the question, right? I always say that we as a clinician, I was also a clinician, was in practice. So we have seen the one side of health, that health has two sides. One is health practice and health research. We always talk about the health practice, but we are not talking much about the health research. It is like a unseen side of the moon, right? But yes, that is also very important, and it is not too difficult to land up into that side. Okay, so my request to all those young people here that please take advantage of that. This is very rare. I see that such kind of zeal and then will of a management and administrative head and the scientists that we can develop. And since they are all, everything is there in place. And this is an era of collaboration, as I say. So you can collaborate, and I am as a community health and as a HDSS, we already have the network of the HDSS sites, Indian HDSS network, we already have the 19 sites. So we are happy to help you, or any help you want, I can do that, and we can collaborate and develop a good evidence for our country, for our people. And once again, thank you very much for this opportunity. My pleasure. Thank you, sir.
Sir, the morning session concludes here, and now we break into group discussion. We will use this opportunity for vibrant discussion, networking, and collaborative ideation. I request Professor L.S. Shashidhar sir to guide us about how he would want to go about this group discussion.
And also the ongoing large projects. Given this background, what I request for each group is to discuss with the visitors and Subbaiah Research Institution experts and scientists and clinicians that what kind of questions they have, very priority questions, what retrospective samples and data they have, what that can be prospectively can collect, and what kind of community initiatives one can start, depending on your own area of research. So we have divided into 4 groups rather, metabolism and nutrition related disorders, then cancer, and cancer is separate one, there's separately one for cancer, and rare genetic disorders, then the cohort study, setting up a large cohort and do public health work, community work, and infectious disease and AMR is another one, I think. And setting up cohort, Anand is here, please you also join us, and Pallavi, cohort, what happens, how you set up biorepository and all, so Lula's building the whole nation. Thank you. And genetics and genomics, Tangraj. So I guess I think we start for an hour or so, we'll have a round of discussions, and then post lunch, it should continue with lunch, the discussion. I think the same group can sit together in the lunchtime also, so that we'll have about 2 hours of discussion. And then some of your colleagues can take notes and make summary of it. We'll get back to each one of the visitors so that they'll provide input and then come out with a roadmap. Okay, thank you. You can also join one of the groups, I guess.
at only one hospital with around 1,000 births every year, it gives us an immense opportunity to use this hospital as a basis to establish how we can proceed the newborn screening program. So what we envisage is that if there is an umbrella human ethics and everything approval, we can take the blood samples of every newborn baby which has been there, collect the genetic material, and maybe and divide the work in such a these are there, and we can actually and for the this can also become cost effective, because if we can club multiple samples together, I'm pretty sure it would be, and it would give us a lot of information. Even if we don't see much of the rare disease in this place, it would at least give an information of the carrier frequency which is associated with it. And since there are multiple number of population which exists in this district, we can categorize these 1,000 birds, geographic wise, community wise. And if we take this study rather from a year, 1,000 birth, and if you think a longitudinal part of 10 years, then you have a great amount of data by which you can make biological deductions, right? And this can actually, apart from rare genetic disorders, it can also address, population diversities. There's a lot of kind of things which can be done with such kind of data, right? And I think, is there anything else you want to add? Handy-goody, sorry, right, that's the thing. The third is, missed the point, yeah, sorry. So the third point is the handy-goody disease. We know that there is a hotspot which is there in the Shivamogga district, and it's in the Malnad community. And we don't know, I don't know, is there no genetic basis even known for the disease, right? So this, if there is a cohort of patients which we can recruit in this region, they can serve as an absolute space to understand the genetics as well as the biochemical basis of the disease. And
basis as known, the sample can be sent to CDFD. I think Ulas will be happy to also help there. So for this program, there's no fund is required, already funded by DBT, so that is taken care of that. Another point is academic. We also thought that we look at, because here a lot of population which are small size, practicing endogamy, that's my interest. Again, funding is not necessary, we can always do. He said that he can send some of his students to come and work on the samples, so that's academic side of this program. Thank you. Any other point?
People there for a field exposure. So now they are taking up a qualitative research, trying to understand not only the disease burden, what are the problems they are facing. As of now, we don't find Andigudu syndrome anywhere in Shimoga, but we are finding it in Chikmagalur, Hodavannur belt. Yes, they are saying it's not there. Maybe one generation is skipped, we don't know. So yeah, it's an interesting area to take up. Our staff are already working on it, so somebody wants to join them, we would be happy to take them. Yeah.
That is being taken care of by a practitioner based on Mysore who's affiliated with SRI. I forget her name.
No, she's from this college
Okay.
only.
Dr. Suma. So she sees a great deal of oral precancerous lesion patients, and we at the lab have developed an in vitro model of precancerous squamous carcinoma based on skin. And it sounded, the detection system that she was describing sounded very much like what we are doing fundamentally in the lab. So they have the perfect model for us actually for our benefit to actually get to a clinical system to look at non-tumor, precancerous and cancers, and with and without a delivery system, patch delivery system of a treatment. So we have a perfect model from us from the fundamental point of view. And from the clinical point of view, they will get to know about some fundamental molecules that we have in the lab that we can detect. So perfect collaboration.
voice is normally loud enough. Okay, that's enough. But the main person I came to meet, so I'm a person who's interested in metastasis. So I will be speaking to the breast cancer oncologist. Unfortunately, he couldn't be here today. So that's why I was telling Shashi, I was outside talking to him most of the time. So we will be meeting on Monday. And the whole hope is that we can get along and, you know, go on to get maybe not warm autopsy programs, because that's very challenging, but even start with metastatic samples, which I think will be very valuable for us. So I hope we can take that forward. That's my bit.
One more thing, she works also in the space of TB and AMR. And recent data from India suggests that Bowen TB is a big problem. In Karnataka, it is around 7%. So we were discussing to have a 2-day stakeholders meeting on how do we do One Health approach for TB, specifically focusing on Western Ghats, where there is human-animal and environment interactions. So we will update you on that. So that is one discussion also which happened on how can we start with TB and then move towards other One Health in Western Ghats. The other thing we were looking at with Dr. Akanksha is during our recent KFT survey, we saw a lot of dead bats. You know, whenever we went to the villages, thick inside the forest, literally bats we saw on the wherever we were walking, dead. So Dr. Akanksha is also interested to explore how do we look at virus analysis in bats. It is a little challenging, but I think we will explore to see what best we can do in that particular area, particularly in this Western Ghats section. Yep. Dr. Sakshi, you want to add anything? Yep.
Western Ghats are actually the hotspot for a lot of emerging and re-emerging pathogens. KFDB is one, and also Nipah. Many, not KFDB, it's not known, but for example, Nipah is known to be transmitted by bats. What we know that the bats themselves don't get the disease, and there is, because they have some inbuilt immunity. So what we were discussing is not only to recognize the virus, but also looking at the bat antibodies, and if we can explore those antibodies and, you know, bring them for diagnostics or for the point of care diagnostics and therapeutics.
TB, and I don't know how long I'll be still with NIB, but as long as I'm in NIB, you know, you'll be more than happy to connect you with people who work on bovine TB. There are a couple of good people, but if there is something that is more interesting, if they find it enthusing, we can launch something together, no problem.
Spoke about what we are going to, you know, what we decided in the round table for AMR and infectious diseases. But there are some points like, you know, like Shraddha mentioned, they have a model for, you know, they have created an in silico testing system with the databases of isolated bacteria, which can be used, you know, for a place like Shimoga to see what is the source, or what is the pathogen pattern, or what is the ARG distinct pattern or characterization. The other thing is acute febrile illnesses, because I work on clinical surveillance, and pyrexia of unknown origin, fever of unknown origin is an area which can be really worked on in this place, especially from both adult and both pediatric samples. It is something which we have also discussed and we are considering.
you keep them in, like once you store them. Because at the end of the day, the owners of the tissue are still the patients. And as soon as the patients ask, we will issue it to them. But we do have capacity to actually make more blocks and keep if we need. That can happen only if we decide that we are going to move in that direction, because additional resources will be required for that and store it in a much better manner, because we require anonymization of that data and we need to create a chain of custody and all that. It will require a lot more of a proper protocol that we need to create, and then also decide which direction we will move in for the biobanking particular actually. So that because it requires a lot of resources, both physically as well as technologically, to store that much information as well as physical specimens. So unless we focus, it will be very difficult to do all of it, because even at the end of the day, it doesn't matter how big we get. All of us have limited resources. Resources are always a limitation. So once we get there, once we get our research question and focus, we could move in the right direction. And it is a long-term process. That's something that I learned. Like ma'am said, that she has been doing this since 2015. So more than 10 years now. And even Dr. Madhura, when I interacted with her, they have been doing it since more than 15 years now, ma'am. Yeah, so it's a very long-term commitment that will require towards biorepository. But those are some of the learnings that I want to get.
I just want to add, also doing a lot of genomics and sequencing, we can also help them actually to organize your antiviral data, which we collect, and have some tools for stewardship, which also have direct patient outcomes. So that also helps you to embark upon more meaningful research, right? Once you have all your data organized in a way, and how to look into it, and you know what to pursue exactly. So I think that funding can also help write a new business plan.
About environmental surveillance, which we have to take that. There are a lot of things which are cropping up right now. And about the biorepository is what we think is the way forward. Of course, it will take a long time. It will not happen in one day. It will take a long time, but definitely we'll have to work onto it. And Dr. Pallavi has promised that she'll do some hand-holding for us along with Dr. Madhura. So it will definitely happen. If we have that inclination towards it, things will happen in the right direction. So we'll have to just work onto it, see how to understand how we can apply for some grants along with it. We can manage. If we really want to do it, it will definitely happen. Yeah. Sir, I just want to ask Dr. Sripathi.
So the three things, right? In the context of infectious diseases, environmental surveillance is going to be the really key, because you may not get compliance patients coming in, but they come only when they're referred by a primary or secondary hospitals. You may not get directly people coming here, because it's a tertiary care hospital and also medical college hospital and kind of stuff. So given that, I think environmental surveillance helps you to be alert, what is new, what's happening. Because of variety of different other kinds of zoonosis, whether it be bats or even from poultry, it's important to continuously do environmental surveillance, and that sort of helps in the community health, and also providing service to the society indirectly, because it may not be visible to ordinary people what you're doing. It will be definitely visible to the public health officials and district officials, because they would know what's happening at the ground and how to prevent an outbreak, right? So I think environmental surveillance is going to be one major one. The second, coming to the cancer or any other metabolic and nutrition related, you have to run a large cohort, which is what we discussed, even for biorepository, right? The cohort, unlike 20-25 years ago when Anand was setting it up, or Yagnik and in Dr. Nehra in AIMS, those times were less mobility of the people. Now, this increased mobility is very difficult to get compliance over, let's say, same individual coming 3 or 4 times in 3 years and giving their blood sample for metabolic analysis, right? So given that, we need to use another intelligent way of just to increase the size of the cohort because it's very likely that at least 2 times you may get from the same individual, may not be multiple times, and there must be some data analytic methods which will help to rationalize how to sort of get the progressive change in the metabolic profile over age, and also seasonal variation, climate change, and all kinds of other problems, right? So I think that's one new method has to be evolved, because I don't think same individual can come for 20 years to give the samples or to collect the data. Of course, you can get some indirect data without collecting the blood samples. Some mobile app, if they are enrolled, they may keep sending some information. I don't know how much will be the compliance. It will be very difficult. The repository also, for cancer, for example, it will not be very large, and particularly if you have some, sort of fresh biopsy sample in the FFP blocks, and it depends on how much space you allocate for room temperature preservation and for cold storage and so forth. So I guess we discussed quite a few points. At least 3 or 4 should start immediately. I can see Rajesh is already on to 1 or 3, and I think adolescent girl one I think is important. I think malnutrition is also higher among them, and it's also a priority, national priority, I guess, right? And so I think there are quite a few. I think each one of them can start in its own way, and funding opportunities are there in ICMR, DBT, DST, and variety of other sources. You want to say something?
So I think, I mean, in terms of immediate thing, I said that we could immediately help with the adolescent girl micronutrient cohort in their the 12 villages that they're already working with, which is we will give you money. We'll give you money to do the cohort, so don't worry. The other thing is the pediatrics might be interested in looking at the body composition of the newborns. If you want, again, there we will supply you all the reagents and we will take care of the cost. Up to 100 is not a problem. You can generate preliminary data and you can go for a bigger thing. But I was talking to the entire CPH people. Your community presence has to be significantly improved, but they're already doing fantastic work, but I think it takes time for you to build that confidence. And also, community participation has to be high, and you need to create mechanisms where the hospital also subsidizes certain costs from the family so that your community presence, which is over. Otherwise, you will have huge resistance even for surveys, but definitely not interventions. Interventions, you'll find communities stonewalling you and you will not get participation. So I think you need to build that trust over a period of time, and it's only then you will have an impact. Otherwise, you will only get people who come to the clinics for treatment. That is not going to be useful. So your community presence has to be good. So I think building your CPH angle is extremely important for everything that we are talking about, whether it's cancer, whether it is AMR, whether it is whatever.
So that's how the Baptist hospital.
Yeah, exactly. So I gave some ideas about how exactly to do. I gave examples from CMC and from Derwent and from other places. So I'm sure that will happen over a period of time.
And from last 1 year,
if you have a good number of primary health clinics, single doctor clinics, who refers patients to you regularly, you set up a protocol for them to collect data on could be simply a mobile app collecting data. That's really valuable. Right now what they do is they simply refer with writing one word, what is the symptom, and then refer to you. They may not give you full details of what treatment is already given, what their impression is, everything. But if they can give some additional details, that data will be very useful for you, because that also will act like a cohort, because the same patients will be coming because the same doctor will be referring to you. It could be small villages outside. Okay. You want to say something?
there are a lot of things coming up. I'm very happy to see all that. And I think I will request all of you all to just have some amount of, because we are in the actually infant stage only, you can say. A lot of hand holding would be required, and we are open for taking things forward in a bigger way, sir.
So for a short one, before.
So I'm seeing that most of the thing we are talking is the idea that visitors are floating and trying to get samples, which is an important thing to do. The other way around also, so we had in our discussion somebody from ENT and somebody little problems like can we know the what antibiotic will not work sooner than 2 days or 3 days. If we know the ERG by surveillance, we will know it can be done by PCR methods within an hour or so. I'm giving an example. What I want to say is that there should be problems which comes from clinician side that we may not be able to solve, but here you have 6, 7 top institutions of the country being represented. They are extremely well funded by your money, and their responsibility is to solve the problems if they can. So I think if you are seeing a problem which you think the solution doesn't exist or is very cumbersome or is very expensive, you should throw as a challenge, and these institutions can try and solve. Being regional, that is one important thing. We should look for some of these global, the very local problems and solve them. It may be diagnostics, it may be certain kind of, maybe at some time making little equipments and those things, and maybe we can catalog those challenges and try solving some of these institutions, and we can, I think that will be one important thing.
It's a good point. They're all used to play, work on grand challenges. Can also be local challenges. Yeah. Okay, so we have Professor Shripati here. He is was a professor of engineering college here, and earlier he was currently also visiting professor at IIT Dharwad. Did his PhD in IIT Bombay. The reason I called him is, one is of course he's the one who connected us to this college. Second is he is very passionate about generating any facts, basically evidence-based, scientific evidence-based solutions to any problems. So first you generate the evidence to understand the problem, and then you try to solve the problem. You're also working very closely with saving the Western Ghats, saving the rivers of Western Ghats. That's why environmental surveillance, bat surveillance that you're all talking about will help in multiple different ways, how because of climate change and human activity, the ecosystem is changing. You generate a lot of scientific evidence which can actually, hopefully, at some point will lead to some policy decisions. Unless we collectively, of course, we should push for that. So, you want to say something?
Yeah, good afternoon. Actually, as Shashi told that I work on environmental issues. My basic profession is engineering, particularly green technologies. Currently, we all of us know that there are a lot of projects coming up in Western Ghats, starting from southern Gujarat to Kanyakumari. In Karnataka alone, there are more than 27 projects are under various stages of implementation. So our worry is how to address this situation. What are the problems, health issues, particularly for human beings? We know that during COVID, lot of discussion took place, zoonotic diseases and etc. But even today, we don't know really how to put it forward across the policy makers. We don't have any data to say that environmental degradation, it may be encroachment of the primordial forest or our water resources really contribute towards the health of the local region. So I appeal to you to, at least a team can take up a project to collect the existing data, maybe of 30 or 40 years. So probably 50 years back there was only one district hospital, and that time also people used to go to Manipal from these places. So there must be a database in Manipal or in our Megan hospital in Shumaga, and later on many hospitals came up in this region. So there must be some already available data. So based on that data, can we come out with some information on what type of changes that have taken place in the environmental side, like water, soil, forest cover, etc. And can they be attributed towards certain types of diseases? We don't know. We have to screen out the type of diseases that are coming up in the recent years in terms of numbers and type, and link whether it is possible to link these type of diseases to the environmental relations. If we can do that, and if we come out with certain concrete data on that, we can definitely talk to our policymakers and show them. Because recently I read one in Popular magazine that plastic has gone into even our brains, and that is leading to dementia and other such cases. So is there any correlation, or are we finding out? Because I am talking about the existing data. We have to collect the data, screen them, and of course we need to know that there are complexities in screening out the diseases and attribute this to certain types of environmental decrease. Particularly in the Western Ghat, we have seen lot of change in the agricultural practices, particularly usage of pesticides, fertilizers. Are they creating any issues with our health related things? So this is just an appeal, because I am a layman as far as your workshop is concerned. I saw one of your goal is to do clinical research as well of societal impact. So probably you may take up this, and if we can get that data, we can use that for our further work. Thank you.
Great. I think, is there a way to get some data, old data, from other places, like government hospitals? Do you know? Is there a way to get some old? Because usually I know almost all the people in the Western Ghat, they either go on that side of the hill or this side of the hill. Those who come this side of the hill come to Shimoga, otherwise they go to KMC, Udupi.
There are some PHCs. The other day I visited, it's 100-year-old health center. There are few health centers which are there since last 60 years. So there will be some data which manual, not digitized. Definitely they will have it. And Megan is one of the oldest hospital also. We can explore, sir, but we need some permissions to get access. Because it's
no, there are standard ethical practices. The permission doesn't have to come from outside.
No, here district
For
level.
Begon Hospital being in the central government, state government, the state district commissioner here has to give approval. There is always a health officer in every district. That person has to give approval. Second is what kind of data is important. In the context of climate change, or because of environmental changes and water, this one, you can actually see certain kinds of data we may have to collect. Like one is of course diabetes, cardiovascular, other one is some infectious disease, diarrhea related one. So that can sort of correlate to.
I think we have data already for last 30 years in NFHS. We can just look at NFHS data, which is currently district level we have access, and see how there is change in last 30 years. It's already done by Government of India survey. If we want to go to micro level, we need to write to them, they'll give permission.
So that will give you a granular, because that I know NHFS district level is not that granular. Because every district is covered national level, it looks granular. But if you want to cover the district level, let's say 2-3 districts of Western Ghats, we will try and get some more data from other hospitals.
In NFHS, we can go up to PHC levels.
I see.
But we need to write to them, they'll give access. Some government agencies, because you are here, they will definitely give access. I think high density, yeah. But at least
PHCs will have high density data.
yes, sir. Maybe this could be the starting point to look at last 40 years NFHS, what percentage has changed up or down in which indicators, and then take it to the district administration saying, look, this is what is happening. We want to do a micro analysis and put a team on what we want to look. Let's not collect data which is.
Let's do one thing. I mean, anyway, you're going to establish cohort.
Yeah.
So you have some cohort study, some new cohort you want to establish. To select the right cohort covering the entire spectrum of the health problems, the old data will be useful.
Yep.
Anyway, multiple reasons why I need some old data.
We'll work on it, sir. Sir is anyway here, you're also there. Anybody also interested? We'll try to work on it. It looks interesting. To start with, I feel let's start with NFHS and then put a policy framework or what exactly we want to get from government, instead of just asking them, we want all data.
Because very old data, which is at least, let's say, if not last 30 years, let's say it's 0 to 20 in the previous one, out of 30-year age one, let's say up to 2015 or something, not 25, then advantage is there are no concerns of the government will come into picture. There will be more, they will say, okay, it's all old data, no political party will get blamed for whatever happened at that time. So they may actually be more open to share that kind of data rather than today's data. Everybody is afraid. Even a pothole is you don't want to do a survey, right?
Correct. We'll work on it, sir. Definitely, it's an interesting thing to do. Yeah.
Okay. To you, final. Thank you all. First of all, thanks from all of us who have come from outside Shimoga. Thank you so much for your hospitality, for sparing the whole day with us. Thank you.
I would like to thank each and everybody for being with us here and giving us so much of inputs. Actually, it's a big enthusiasm for our own researchers to take things, our own faculties and all. And I request our faculties also to follow. We should not end here. It should move forward. Each one of you who ever interacted with each person from our visitors have to take their vertical forward and see something comes out of it, not just let it off here today. And we should have follow-up meets. And also it is very nice to have you all from, I don't think I could have managed without Dr. Shashidhar's help to have so many of you all under one roof. And we will also visit your institutions to get a better insight into how we could also progress, and if possible every 3-6 months, if not offline, online meets or something such that we can take some, each projects what we have decided today can be taken forward in a little bigger. So I will ask one of my faculties to make a minutes of the meeting of what things have cropped up out of this and share it with you all, and then so it'll be a reminder call, okay, we have to work on to these things forward. And again, I'd like to thank Shashidhar sir for, mentoring all this and taking this meeting and making this meeting possible. Also, I thank my faculties also, who have actually on a Saturday, actually Saturday is a half day here, but then they have till 3 o'clock and very enthusiastically meeting everybody, taking inputs from all of you all and trying to take it. So we just hope for the best and see what best comes out of it. But definitely we want to develop a cohort. Subbaiah cohort is what I was thinking about, but then again we require a little amount of hand holding from all of you all. So, and of course the rural development program of actually very, I met sir yesterday in Royal Orchid, Dr. Anand, and he has a little incentive of coming to Shimoga often because his son is going to study in our college. So every time he comes, I think we can have a small meet. But then we can arrange some at least 3-6 months. The way forward, we thought we'll have a AMR workshop also if possible. That's what Dr. Vijayshri also suggested. We'll see how to take it forward. So there are many things coming up. Now we'll see. Just hope for the best what comes out. I was talking to Dr. Rakesh regarding how we want to send our students. They have an elective of 15 to 20 days. So 15 days, no? So, 15 days of third year clinical postings to NCBS. Similarly, I thought we could send to TICS also. So sir agreed. So that was another thing which we could send, so that they get a little research exposure and they can again help our faculties to take it forward. So I'd like to thank all of you all, and we'll want you all to join us for dinner in the evening at Royal Orchid only, no?
Translational research is the only way ahead for us. Otherwise, we have no role as practitioners and who have data, who can share data. So I have different acronyms and abbreviations for all the members who have attended here. So NCBS now for us stands not National Center for Biological Sciences, but Noble Champions Building Solutions for India. I hope you continue and leave a legacy there. I was very impressed with Dr. Ullas. I couldn't have a T with you. But anyways, so the most interesting thing was malnutrition. So I had discussed this with Dr. Shashidhar. So we were, when undergraduates, dealing with MAL nutrition, malnutrition. Now it is MALL nutrition. So you know what malnutrition is? MALL nutrition is ultra processed foods. And so from mal, we have come to malnutrition. So it was fascinating. So for you, Arunda, I think because of the amazing rural work, rural research that you're doing, I thought amazing researchers united for making difference across India is what I felt. And I was very impressed with Mishra sir when you said, when Mishra sir said that rural people or rural health sectors should be within the ambit of advanced research, and from that would actually enable what is called as bench to bedside. So I think TIGS stands for Talented Indians Generating Solutions for Everyone. And then we have, I've had a lot of good discussion with Dr. Iyer, and Mansi is already in our team. So I couldn't have any discussions with the CCMB, but what I felt was CCMB stands for no longer CCMB in the original format for us. For us, it is Creative Champions Making Breakthroughs for India. So, for us, TRIF is no longer TRIF. It's Talented Indians Researching Future Generations. So with this, I just sum up whatever has happened. And I hope we will be of some use to you all, because our team was really apprehensive and anxious to host you all. Because you seem to be one of the, you were all talking of cohorts. You probably are the most creamiest of the cohorts in research. So we were all petrified, but I hope it has not gone too bad. And thank you all so much.